Okay, so hello everyone. My name is Nicholas Alegre, and I just want to start off with saying thank you for coming to my presentation here at the Undergraduate Research Symposium. So to start off with, I'm going to be talking a bit about malaria. Now, malaria is one of the worst diseases ever known to man. The majority of the malaria cases are caused by parasites of the Plasmodium family, especially the Plasmodium falciparum, which is responsible for the most deaths by malaria. Now, in 2019, it was estimated by the World Health Organization that there were over 400,000 deaths caused by malaria alone, disproportionately located in the India and Africa regions. Now, typically when a person has malaria, they're going to be treated with an anti-malarial drug known as artemisinin. However, in 2008, it was found that artemisinin-resistant strains of malaria started to show up in Cambodia. And so because of this, being able to find new and innovative ways that we can stop this disease is imperative to saving so many lives. So one of the main things about malaria is that it has a very distinct life cycle here. As you can see on the right, you can see the parasite enters into the human host from a mosquito vector, where it enters into the bloodstream, and you have this asexual reproduction stage with these early ring stages, a more mature trophozoite stage, which will turn into a schizont, which will rupture into small little merozoites that continue the cycle until the parasite leaves to reproduce. Now, one of the most interesting things about the malaria parasite is that it actually has a very heavy reliance on what is known as chromatin structure in order to control which of its stages it's in. So if you look at the bottom here, you can kind of see this is a chromosome of an organism, right? In this chromosome, you have DNA wrapped around different protein subunits known as histones. When you have this closed off chromatin, the necessary transcriptional machinery to express genes is unable to bind. However, in contrast, these open regions of chromatin here, you can see that this transcriptional machinery is capable of binding and activating these genes. And so because of this, if you look at different ways that you might interfere with Plasmodium's chromatin structure, you can find ways to interfere with its life cycle and stop this disease. So in recent years, there was a new method developed known as the assay for transposase accessible chromatin, which has been shortened down to ATAC-seq. Now, essentially what ATAC-seq is it's a method that allows you to map the chromatin landscape. So utilizing a mutant enzyme known as transposase, this enzyme can cut and tag accessible regions of DNA. So if you see here, we have a diagram of the open chromatin regions here. This transposase enzyme can cut and tag these locations in a process known as tagmentation. You'll take these fragments and amplify them with a process known as PCR and sequence them. Because these open regions of chromatin are more likely to have this transposase bind to them, essentially what you're left over with is different reads, and these open regions have more reads. So if you look at the different reads that you get from this ATAC-seq protocol, you can essentially create a map of the chromatin to see which regions are open and which regions are closed. In addition to this, though, this process has also been refined down to the level of individual cells. So you can actually perform ATAC-seq on individual cells in a process known as single-cell ATAC-seq. Now, the original goal of this project was to take the nuclei extraction step of the ATAC-seq protocol and try to optimize it to work with Plasmodium falciparum. As you can see here is some kind of altered lysis times of this nuclei extraction. However, unfortunately, as I'm sure you all are aware, we had the COVID-19 pandemic. And so because of this, access to a lab wasn't always readily available. So instead, this new project wanted to focus on looking at the relevant single-cell ATAC-seq literature in order to better understand it, especially taking a look at some of these single-cell platforms and then some of the advantages that single-cell has over the bulk ATAC-seq. So starting off with the platforms, there are many, many types of different platforms that exist. However, in many of these papers, they kind of boil down to two main types. On the left here, you have what is known as split and pool combinatorial indexing, shortened down to SciATAC-seq. So in this method, you'll take these nuclei, put them in a 96-well plate, along with your transposase enzymes. These transposases will mark these nuclei with different tags. So you'll take the split step, then pull them together. After this pooling step, you'll split them once more into another 96-well plate where you'll have more adapters. So then using the technique PCR, you can amplify these fragments, and eventually you'll be left over with different fragments with a unique combination of two different markers. And so that's how these cells can be resolved at the single-celled level. So kind of the main advantages of this technique are it has a relatively low read number of only 2,500, but it has a relatively large cell number of around 1,500 to up to 17,000 cells potentially. And finally, another advantage of this method is that it doesn't really have the need for expensive equipment that you'll see in a bit. So in contrast to this, the other main type of single-cell technique method utilizes what is known as microfluidics. So in this method, you'll take a specialized machine with this microfluidics chip, and this chip has different chambers where you can take your isolated nuclei, which have been transposed, in addition to gel beads with a unique molecular tag. So using this machine, you can take these beads and these nuclei, immerse them in oil, and get individual nuclei with single beads. And then using PCR, you can amplify and integrate this tag into the nuclei. So in the end, your fragments will each correspond to the unique gel bead that they were with. So this is the microfluidics method. This method is known for its very high number of reads, up to 70,000 in the early microfluidics, but it had the disadvantage of a fairly low cell count of 96 initially. But in recent years, microfluidics has become the more increasingly type of popular ATAC-seq protocol. As you can see, because we have the advent of the commercially available 10x genomics chromium device. So this device is able to take cells of up to 10,000 cells times 8 lanes per each of these microfluidics chips. So you can see it has a very high cell number with the high read depth of microfluidics. So now that we've talked about some of the different platforms of single-cell ATAC-seq, we're going to go a bit more into some of the advantages that the single cell has over bulk. So normally when you perform ATAC-seq in bulk, the chromatin landscape that you're discerning, it's an average of all the different cells that you have in your sample. So if you have any rare cell types or different mixed populations, you're not really going to see their signals. So in contrast, when you have this single-cell version, you have the advantages of being able to take a mixed population of cells, identifying their different ATAC-seq signals. You can also identify regulatory elements that are unique within individual cell lines, such as enhancers, promoters, or different transcription factors. And then finally, you can also use single-cell ATAC-seq to track the development of individual cells as they go from stem cells all the way down to fully differentiated cells. So to start off with here, you could see one of the biggest advantages, as mentioned, was sorting these mixed populations of cells. So researchers using single-cell ATAC-seq were able to take different cells of different species, the human GM12878 cell line, and then mouse cells. Since each of these cells should be expressing their own unique genes, they'll have their own unique ATAC-seq signature. And so by discerning these different signatures from this mixed population, they were able to determine that more than 90% of the cells that they found could be grouped to either the human or the mouse cell. But then they also went even further, using ATAC-seq on cells of the same organism, but different cell lines. So they took the HEC293T cell line or the HL60 cell line and did it again versus the previously mentioned GM12878. And so taking these mixed populations, they found that more than 95% of their cells were able to be mapped to either one of these groups. And then finally, they were actually able to go even further, utilizing single-cell ATAC-seq to see different cells of even the same cell line. So once again, the previously mentioned GM12878, they did ATAC-seq and were able to find that this cell line had 4 major cell subgroups based on their ATAC-seq signatures. And another example of this can be seen when researchers took pancreatic islet tissue. Using ATAC-seq on these cells, they were able to identify unique ATAC-seq signatures for the alpha, beta, or delta cells. Now, one of the things to note here is that tissues are often made up of these mixed populations of cells. So you can perform ATAC-seq on these individual samples and find the individual tissues and rare cell types. In addition to this, in diseases like diabetes, for example, it's often known that these beta cells, particularly in the pancreatic islets, might be responsible for diabetes. So this just highlights some of the uses of ATAC-seq besides research. The next main advantage that you have is the ability to identify different regulatory elements within cells. So if you take the chromatin structure based on single-cell ATAC-seq of these two cells, one region of the chromatin might be a bit fairly similar to each other. So you have a uniform ATAC-seq signal between these two cells. So this kind of hints that these regulatory elements are fairly generalized. But in contrast, if you look at another region of the chromatin, you might see this variable signal between these two cell lines. So that kind of hints that these factors are specific to the individual cell lines. As you can see here, this is some data from the researchers where they found that certain factors here were very amplified in the variability, and then you had different buffers. So you can see that some regions are more specific and then some are more generalized. However, this can also be used to kind of see how these factors interact in a cooperative or competitive manner. For example, if you look at one binding site on the DNA that's available to factors GATA1, GATA2, TAL1, or P300, if this binding site was available to these factors, it showed very high variation. So that kind of hints that these factors might kind of compete for each other to try to bind to that location. In contrast, if you look at another binding site available to GATA1, June, or CEBPB, you could find that it has very little variation. So even though this binding site is available to all these different factors, these factors might interact in a cooperative manner, which is why you see a more uniform signal. Finally, one of the last main advantages of single-cell ATAC-seq is the ability to track the development of cells in an unbiased manner. So when you look at different cells, when they differentiate, starting from the stem cells to the fully differentiated cells, each of them needs to express different genes. They'll have different chromatin structure. So performing ATAC-seq, researchers were able to take cells of the blood and bone marrow, perform single-cell ATAC-seq, and group their signals into different clusters. Then they compared the signals of these different clusters, starting from the plasma B cell. They found which ATAC-seq signal was most similar and found that it was probably the previous step in differentiation. So using single-cell ATAC-seq, they created this known pathway in reverse to kind of highlight how single-cell ATAC-seq can understand the development of these cells in an unbiased manner. Finally, I kind of wanted to close off on a bit about ATAC-seq and malaria's use. So currently, there is no single-cell ATAC-seq with malaria. It is still at the bulk resolution. But some of the research that has been done with bulk ATAC-seq in malaria highlights just the uses that we can use with single-cell ATAC-seq. So to start off with, we have some validation here. So they observed previously established chromatin changes associated with malaria. For example, you see more accessible DNA in the late trophozoite stage and the early schizont stage of the malaria parasite. So looking at these nuclei of these malaria parasites, you could see at the trophozoite life stage, you have these factors in this repressive cluster here. However, when you switch to the schizont stage, these factors move out of this repressive cluster and are able to be transcribed. So that just validates how ATAC-seq can be used with malaria. But with research, they were able to identify new regulatory elements in the malaria parasite's chromatin. So for example, they identified open regions of chromatin using bulk ATAC-seq, and they knew previously from other research that a transcription start site is usually associated with certain histone modifications, the H3K9 methylation or the H3K4 trimethylation. And then in contrast, these enhancer-like regions are usually associated with this histone H3K4 methylation. So this just signals how ATAC-seq can be used in a parasite like malaria in order to identify how its chromatin is regulated so you can develop new anti-malarials to fight it. So in conclusion, I just wanted to leave off that single-cell technology is essentially the future of molecular biology. Many of the advantages that I mentioned, this deconvolution of mixed cell types, identifying these individual regulatory elements in cells, and then tracking development in unbiased manners. These are just some of the advantages that it has for bulk techniques. And then there's a wide range of applications from research to medicine. The possibilities are endless. So then I'd just like to give my thanks to my faculty mentor, Dr. Corinne LaRocque and her lab for helping me with this process, as well as the University Honors Program for allowing me the opportunity to present here today. and also for doing this project, especially the director, Dr. Richard Cardullo, and my advisor, Ms. Latoya Ambrose. And I guess now I'd just like to leave it off for any questions if anyone has any.
All right, and go ahead and stop your recording there.